Annabelle Malinowski, and as Dr. Frederick mentioned, I'm going to be discussing with you the evolution of genomic anomalies in subcluster A5 Mycobacteriophages. So the first question we need to answer is, what's a mycobacteriophage? They are a subcategory of a group of viruses called bacteriophages. Bacteriophages are viruses that are going to infect bacteria, and mycobacteriophages are those that are going to infect the genus Mycobacteria. What's interesting about them is they have two different life cycles. The first is called the lytic life cycle. What will happen is our phage over here will come and insert its DNA into the bacteria. The phage DNA will then hijack the DNA replication, transcription, and translation systems of its bacterial host. It will multiply, assemble more viruses, and then burst outside of the bacteria, thus killing the cell. The second type of life cycle is the lysogenic cycle. What the phage will do is it will insert its DNA into the genome of its host bacteria. So the two will essentially become one, and then as the bacteria replicates, the DNA of the virus will replicate with it. But whenever it undergoes stress, then the virus knows, okay, I gotta get out of this bacteria or I'm gonna die too. Then it will go from lysogenic to the lytic life cycle and then burst, thus killing the bacteria. And what can happen during the lysogenic life cycle is an example of mobile genetic elements. These are sections of DNA that can switch from the virus to the bacteria and vice versa. Now on to the materials and methods. So LeTourneau University is involved in the C-Phages program, which is run by the Howard Hughes Medical Institute. In the first semester lab, we actually found our own viruses. And so in the second semester lab, we're actually annotating and examining the genome of the viruses that we found. So my lab partner and I were working on a phage named Dublin. So this is the lovely, lengthy process that we went through, and I'll explain it step by step. The first step was using DNA Master. Now, DNA Master took the nucleotide sequence of Dublin and then said, hey, there are 86 genes in Dublin, and we know where it starts and where it stops. So this program was sort of the motherboard of the entire annotation process. Here we were able to take notes on every single gene. So what we did is like, okay, it gave us certain starts and stops, but we want to go ahead and double check these. So we had different tools we used for that. The first tool we used was Genomic. It would, these areas here indicate where there's coding potential. Coding potential means that that section of the DNA is actually going to code for a particular gene. And so all these numbers here are the coordinates of the DNA. So we would look at the coordinates here and then compare those to what DNA Master had already called to see if they matched up. The second tool we used was very similar. It was called Starterator. All these different lines list the different potential start codons for every single gene. So here is Dublin. This is for the last gene, 87. So this one says, this is the most likely start position of this gene. But here are all of the other ones that it could potentially, again, we would check this against what Genomic and DNA Master had already called. So the reason we had to do this first was because if we were going to use the amino acid or nucleotide sequence, we had to have that firmly established at the beginning. So our next step was to figure out what the job of each gene in the genome was. So one of the first tools we used was FAMRater. Now, each one of these individual boxes is a gene. The reason they're different colors is because each gene falls into a different category. And all these regions say, hey, this area right here is very similar to this area in this other genome. So each of these different tracks is a different genome for a different phage. And in some of these, what FAMRater will also sometimes have little yellow rectangles inside of it. They'll tell what the job of that particular gene is. So what we can do, we can go back to DNA Master and get the amino acid sequence for each gene. Then we take it to a website called PhagesDB and do a local protein blast. So we put in the amino acid sequence, hit the blast button, and it will give us something that looks like this. So this area at the bottom says, okay, We know it matches up, that amino acid sequence matches up with itself, shocker. And here are some other phage parts that it will be homologous with as well. So these values are the E values, and the lower the number, the stronger the homology. Homology is just a fancy biological word for being the same. And the red area here indicates that there's very strong homology, as with those. HHPRED is another tool that does the same thing, but it's going to compare the amino acid sequence to a larger range of organisms. So at the very top of the screen, it will look like this, saying, you know what, the amino acid sequence you put in, it matches very closely with this. As you scroll down the screen, you will see a very lengthy list of what it predicts your particular gene to be. And numbers 2 and 3 are very promising. They have very high probability and very low E values. So it looks like the particular gene we blasted is going to be some sort of terminase. And then I can click on number 2 and then scroll down, and it will give me more of the specifics for that particular gene. We can take these notes from HHPRED, the BLAST-P, and NC, and then FAMRater, and put that in the annotation notes inside of DNA Master. Later on in the process, we were curious as to how different phages were related to each other, so we made this beautiful phylogenetic tree, but we'll get to that a little bit later. So throughout the annotation process, we had to abide by certain guiding principles. These just basically listed what we would expect to see throughout the annotation process. And number 3 proved to be the most useful. The gene density in phage genomes is very high, so genes tend to be tightly packed. Thus, there are typically not large non-coding gaps within genes. And elsewhere within the guiding principles, it also states that we should expect no more than 80 base pairs between genes. But during the annotation of Dublin, we found an anomaly. So this picture shows the extreme right-hand side of phage Dublin with its second to last and final genes, with no auto-annotated genes called in between it. Ladies and gentlemen, this is an 863 base pair gap. And according to the guiding principles, this should not occur. Our research was centered around figuring out how and why this happened. So the first thing we did is we highlighted these different regions that looked like, you know what, there could be a gene there, and there. We took those amino acid sequence and then put them into phages DV, BLAST-P, to see if there were any homologous proteins. We did not find any. Then we went back to GeneMark to see if there was any coding potential within the region. Again, there was none. So at this point we asked ourselves, okay, does this last gene even exist? Because it would be much more easy for the processes of annotation to say that the genome ended here. But when we went back to GeneMark, surprise, gene 87 does indeed exist. So that means we're stuck trying to explain this 800 pair gap. So we took a step back and asked ourselves, okay, so that's just for this one phage. How does it compare with the rest of the phages in its own subcluster? This table shows the extremely strong homology or similarity that Dublin shares with all other cluster A5 phages. Important to note, Thea is probably the most similar phage to Dublin. It has a 95% identity. Identity is just means similarity as well. So their genomes are 95% the same. So we made a FAMRater map comparing Dublin to Thea, Kuko, and Little Cherry. And look at that. Not only does Dublin exhibit this gap, but so does Thea, Kuko, and Little Cherry. So, based on this, the 800 base pair gap is not just some anomaly in a single phage, it's rather a hallmark of subcluster A5. So from here, we can ask ourselves, okay, so that's subcluster A5, and cluster A has a lot of subclusters. What about the rest of cluster A? So we decided to pick phage Natsiatali from subcluster A2. As you notice, it does not have this large gap, and there are genes that are coded in the same position. So we thought, okay, Naciatali and Dublin are probably evolutionarily related. After all, they're both in cluster A. So could it be possible that somewhere along the evolution of these, there might have been some sort of mutation in one of these two spots that would have stopped the coding of what should have been these genes? But before we started testing that, we wanted to make sure that Naciatali and Dublin were actually similar enough to draw a comparison between the two. So the first thing we looked at is their ratio of 4 traverse genes. Green means they are translated from left to right. Red means they're translated from right to left. Reverse, forward, they have the same ratio. And then we also took the genomes of both phages and then using another software called NCBI Global Line, compared the two. They found a 56% identity between them. Now the threshold for comparison is a 50% identity, so 56% was enough to go with. So what we did, we went back to Famerator, and then for all of these different areas where they overlapped, we highlighted the sequence, and then this gave us the nucleotide sequence for both Naciatali and Dublin. We took the nucleotide sequence for Dublin and put it into a phages DB blast N to see if there were any potential transcripts. Again, and not surprisingly, we did not find any. So at this point we asked ourselves, okay, how closely are Dublin and Naciatali actually related? So this is the first phylogenetic tree that we created. We have A5 phages over here, Kugo, Little Cherry, Dublin, Thea. Then we have Naciatali. And each of these areas right here represents a common ancestor. So the common ancestor of Dublin and Naciatali would be there. These are outliers, meaning they are in very different clusters, but in order for the Genius software to work, you have to have some outliers. Now, when we presented this research at the C Phages Symposium, someone asked us, hey, is A2 actually the closest related subcluster to A5? But when we went back and made another bigger phylogenetic tree, we found this. A2 is actually a little bit further away from A5 than we first thought. The closest subcluster is actually A8. Can we still draw a comparison between the two? Sure, they're common ancestors in the middle. So we asked ourselves, okay, we have very large non-coding gaps in A5. Are those in A8? So we went to Famerator and took a look at some A8 phages. And interestingly enough, not only are there large non-coding gaps, but they're in pretty much the same position as in their A5 cousins. So we have large non-coding gaps here, here, and here. Again, indicative that they too share a common ancestor. So what can we conclude from this? We know that a phage's DNA can mutate as it jumps from bacterial host to bacterial host. The DNA can be mutated by either point mutations, insertions, or deletions. And we have strong reason to believe that this is most likely what happened in phage Dublin between Naciatali and itself. And we also know that all A5 phages have this gap that does not have any coding potential. So in terms of future research, we want to take a step away from the strict bioinformatic analysis. What we want to do is create primers that align with the second to last gene, 86, and then gene 87, which is the last gene. And then each of those regions in the 800 base pair gap where we expected to see coding potential. So we'll create primers for those, and then using reverse transcriptase, heat-resistant DNA polymerase, and nucleotides, create cDNA. Once we create the cDNA, then we can do RT-PCR to go ahead and amplify that and then see if we have any potential transcripts. Now, of course, I did not do this research alone. Madeline McAllister, another LeTourneau student, was the one who actually discovered phage Dublin and actually isolated, characterized, and purified it. Our thanks goes to the Howard Hughes Medical Institute for accepting LeTourneau University into the C Phages program. Kudos to my partner Junia for putting up with me throughout this entire annotation and research process. And of course I want to thank Dr. Balrain and Dr. Frederick for their guidance throughout the entire process. Now this is a fun family photo. A number of weeks ago we were fortunate to attend the Texas American Society of Microbiology Conference. We did a poster presentation competition. That's Junia and I, and Nicholas Leonard, one of our classmates, actually got second place for his poster in the Mycobacteriophage category. So we got to bring home the bacon for LeTourneau. And we had a blast with the family. And with that, I'm open for questions.
What questions do you have for Annabelle?
So with it being what was it called again? Mycobacteriophage. So it is a virus that specifically attacks bacteria?
Yes.
And specific bacteria.
Yes.
Did you check for possible tRNAs in that large gap that you had?
We did not, but that'd be really cool to see if there are any.
Did you
Yes.
not? You didn't run
We
a
did
tRNA scan or
run tRNA
Aragorn?
scan, and the only tRNA it detected was the one that DNA Master auto called between genes 4 and 5.
And Aragorn as well?
Yes.
So there
So it only-
shouldn't be any.
So it detected one in Alpha. Yes.
Is Aristotle lysogenic or
Aristotle? Don't you mean Dublin?
Dublin, yeah.
I am not exactly sure.
Because if it's lysogenic, isn't it possible that that insertion is insertion from the bacteria, right?
Yeah. So it's probably most likely lysogenic.
Okay. Did you blast those genes in HH pred to see the matched Mycobacterium or something?
Which genes are you referring to?
Because you had all these hypothetical genes, right? You put those in HH pred.
Those HH pred with like these nucleotide
You
sequences.
get a bunch of hypothetical genes farther back, right, on the 6' translations. Yeah, those.
Oh yeah, those.
Did
We
you put
did.
those in HH pred?
We did not, but we did BLAST P and then the NCBI BLAST P. But HH pred would be a good one as well.
Yeah, because that brings up, I think, more.
Yeah, it compares it to more organisms,
So
right?
you did do NCBI blast P?
Yes, yeah, we did NCBI
So
BLAST P.
that would pick up anything that's an HH pred,
Right.
except for possibly domains.
Any other questions?
You
Yes.
mentioned the function of a gene was terminase. What does that do?
Terminase, let's see. Terminates the genome. Thank you, Junia. Thank you. Terminates the genome.